Just waiting for a few more minutes, after which I think I can start with the live session. Just a few more minutes if any other people are joining. Okay, so I think we can start. So, hello and welcome everyone to the live class session of week 9 on the course Experimental Biochemistry, which is being taught by Professor Shagata Dasgupta and Professor Shomude of IIT Kharagpur. I am Srijan Sen, and today I will be taking your week 9 live session on the course. So today I will be discussing on the characterization and the isolation of DNA, and also discussing with you people a few questions which will help you in solving your assignments and all. Okay, so I think we can start, and I'm sharing my slide. I guess my screen is visible. Can you please confirm if my screen is visible to you people? Okay, that's good. Okay, so I think I can start. So just as I said, so today we would be discussing, so in the last class we discussed about the isolation and characterization of proteins, and we have been dealing mostly with the different experimental techniques and all about this. So in today's class we would be handling with the nucleic acids part, that is your characterization and isolation of your DNA, somewhat a part of your RNA, and mainly focusing on the DNA. So before moving on, so first we need to know the different types of biomolecules which are present. So biomolecules can be classified into four parts, mainly your carbohydrates, which includes your sugars. Some of the examples of your sugar means you have three carbon sugars, then your four carbon sugars, then five carbon sugars, that is your pentose, triose, hexose. So these are different sugars present. In your six carbons you have glucose, galactose. In your then you have fructose. In your five carbon you have ribose. So these are the different types of carbohydrates which belongs to the first category of your biomolecules. The next one is your lipids. So lipids, we know that the plasma membrane is made up of the protein, lipid, protein bilayer. So we have heard of this lipids term from the plasma membrane. We know what are the components of it. So this is one example of a lipid moiety where your So this is one example of your lipid moiety, which is also known as your fat. So this has a hydrophilic head, which mostly stays outside in the company of your water, and then the inner part is your hydrophobic tail. So this forms the core of it. So the hydrophilic head, which is water loving, likes to stay in the surrounding or outside where you have proximity to the water molecules, and the hydrophobic tails remains in the inside of the part of it. So in the hydrophilic head there can be different moieties, like your it is the glycerol. Then you have with one of the hydrogen one of the hydroxyl group is replaced by your phosphate, and then choline. So this is a phosphatidyl choline, and then this is the hydrophobic tail, and so this is one of the structure of your lipids. And so this is how the protein lipid protein bilayer is being formed, the PLP bilayer. The next category is protein. So we have talked a lot about proteins and the building blocks of proteins, and how are these proteins being made, how can we isolate these proteins and purify these proteins, what can we do with the pure proteins, the different applications and all. So we have discussed about it, the different techniques used for the purification, and we have talked a lot about proteins. Then we spoke about enzymes, which is another class of proteins which actually enhances your reaction without being directly involved in the reaction and thereby reducing its activation energy. So we have talked about all of these. The last category includes your nucleic acids, which mostly includes your DNA, RNA, ATP. So DNA is your deoxyribonucleic acid, your RNA is ribonucleic acid, and then you have ATP, adenosine triphosphate. So what are these nucleic acids composed of? So mostly they have the sugar, which is your ribose sugar, and the phosphate group is attached to it, followed by a base. So these base are of four types, and what are the types I will be discussing later. So the main composition of these nucleic acids mainly includes your sugar, phosphate group, and your a base. So these are called the nitrogenous bases. So, but this is a monomer. Actually, your DNA, RNA stays in the form of polymers. So they are linked by some diphosphate linkages and phosphodiester bonds and this and all. So there are also different hydrogen bonds involved in it, and how many hydrogen bonds for each of this I will be talking in my later slides. So all this composition, that is this monomer, we can call this monomer as nucleotides. So there are two terms which we need to keep in mind. One is nucleoside and one is nucleotide. So nucleosides are the ones in which the nucleic acids are composed of the sugar and the base, but the phosphate group is not attached. But your nucleotide is the one where we also include the phosphate group. So nucleotide has a phosphate group, nucleoside does not have a phosphate group. Okay. Now, what does these nucleic acids do? So, nucleic acids are important in storing and transmitting of hereditary information. So, among these nucleic acids, we have two. One is your DNA, which stores information in the form of a genetic code. So, we have all heard of the codons and all. So, these are important in storing of informations, and then the DNA starts replicating its own replication, then undergoes transcription to give us some message. The three types of RNAs are there. We know it: messenger RNA, ribosomal RNA, and the transfer RNA. So what this RNA does? So the messenger RNA is involved in carrying the information to the proteins, to the synthesizing machinery, and then the ribosomal, the rRNA performs the functions, and your tRNA is important for transferring of informations in the form of proteins via your translation machinery. So your DNA stores the information in the form of a genetic code, whereas your RNA is important in carrying the information to the protein synthesizing machinery via your translation. Okay, so we have previously heard about your DNA to RNA is known as transcription. We have heard of DNA replication, and then RNA to protein is a translation. Okay, so in the structure of DNA and RNA, we mostly have the phosphate group, then we have the pentose or the ribose sugar, and the nitrogenous base which is attached to it. So this phosphate group structure mostly remains constant, and because of this phosphate group we call these as nucleotides, and these phosphate groups are important for giving a negative charge to the DNA. So mostly we have heard that DNA, it's deoxyribonucleic acid. So why is the DNA or RNA an acid even though it has a nitrogenous base? So we have heard that bases are positively charged. So how come it's an acid? So it is an acid because of this phosphate group being attached to it. Okay, so because of this is involved in giving the negative charge and making this DNA and RNA acidic. Okay, so these are the two structures of your DNA and RNA. So both contains your 5 carbon sugars. So here we have the OH group. So this H is actually involved in, so this group is actually replaced by your nitrogenous base, and in here this OH is replaced by the phosphate group, and then you get the two structures. Okay, so what are the differences between them? So what are the differences which we can find out between this DNA and RNA? Can any one of you tell me what is the difference between DNA and Hello, am I audible? For how long was I disconnected? I don't know actually. How much have you heard? Okay, so how long was I disconnected? How far have you heard the lecture? Okay, so did I speak about the DNA and RNA structure? So I was just asking a question that what are the differences between a DNA and a RNA? What differences can you just tell me between them? Not maybe all of them. How much can you remember? That is the question which I was asking. A few maybe. Okay. So just if you can tell me what are the differences, the structural, functional, stability wise, anything will do it. Anything can you recollect about the differences between your yes, that is true. So the stability of the RNA is less than the DNA. Any structural difference can you tell me from the structure? So these are the structures of your DNA and your RNA. So any structural differences? I was telling this OH group is replaced by your nitrogenous base, and this OH group is replaced by your phosphate group. So these two are similar in both the cases. Any other differences which you can tell me? Okay, anyways, so one of the differences from the structure which you can tell is that your RNA has OH group in its 2' position, whereas it is hydrogen in the case of DNA. So one is this difference from structurally you can tell. If you tell about the differences in the location of a DNA and the RNA, so DNA is mostly found in your nucleus and your nucleoids, whereas you can mostly find your RNAs in the cytoplasm. As previously discussed, that your DNA is important for storing the information in a genetic code which performs different functions, which is important for your development, then reproduction, etc. Whereas your RNA carries the information to the protein synthesizing machinery, and it is mostly involved in your protein synthesis, and sometimes it plays an important role in your gene expression. Then another important difference is that of the nuclear means of the bases, the nitrogenous bases which are found in your DNA and RNA. So it is that in your DNA you find A, T, C, and G, whereas this T, that is the thymine, gets replaced by uracil. And then as you already mentioned, that your the stability, the RNA is less stable than your DNA, and this DNA attains a double stranded structure, and it exhibits double helix structure, whereas your RNA is mostly single stranded, and sometimes it folds back upon itself, forming different secondary and the tertiary structures. Okay, so these are some of the basic differences between these two moieties. Okay, we just need to keep them in mind. And this is the structure of your nucleotide, having the phosphate group, the pentose sugar, and the nitrogenous base. So nothing too much to speak about this. Okay, So these are the different nitrogenous bases we know. In your pyrimidines, we have your cytosine, then your thymine, and your uracil. So thymine is mostly found in your DNA, and your uracil is mostly found in your RNA. Whereas in the purines, we have two. One is your adenine and the guanine. So your adenine forms hydrogen bonds with your thymine in DNA, or adenine uracil in RNA, and your cytosine forms hydrogen bonds with your guanine. So these are some of the structures you can remember, because there can be questions or MCQ questions, like you are either given the structure and asked to tell which of the nitrogenous base is this. Like you are given a structure of say thymine, and then you're given four options, and then you're asked to find out. So then you have to remember these structures. Or maybe you are asked tell me the structure of, or take the structure of cytosine, or say adenine, and then you are given four different structures. So there can be questions like this regarding the structures of these nitrogenous bases. So first of all, you need to remember which of these lies in which category. So the pyrimidines includes your cytosine, thymine, and uracil, and your purines includes your adenine and guanine. And then you have to remember the side chains. So there is a link, so you can remember it in this manner, like your cytosine has a CO, and your cytosine is involved in hydrogen bonding with your guanine. So your guanine, so this oxygen will form hydrogen bond with this hydrogen. This NH2 hydrogen bond, this hydrogen bond forms with this oxygen, and then this N forms hydrogen bond with this hydrogen. So there are three hydrogen bond. So like in this manner, you can actually tell as to how many hydrogen bonds are formed, and which groups are actually involved in forming of these hydrogen bonds. And in this manner also you can tell as you can comment on the side chain groups, or the groups present in the sides of these pyrimidines and the purines, and the different names of it. So there can be questions on these structures. Okay, so as a unit, these three components are basically involved in making up a monomer. So this is the monomer. So what is a monomer? So it is an individual unit that acts as the building block of large biological molecules. So monomers are nearly identical units. So now what we see over here is the DNA and the RNA, which is basically a polymer. So it is polymers are large molecules which are made up of these small identical monomers, like you can think of a chain. So these polymers can be different. There can be uh, different types of monomers which are actually forming a polymer, like your homopolymers, heteropolymers. So there can be even different monomers forming a polymer, or even the identical units forming a polymer. So for a DNA or an RNA, it's the identical. Now you cannot say identical units basically, because the nitrogenous bases vary over here, but the ultimate structure of it, the backbone of it, remains more or less similar, forming polymers. Okay, so probably this is a short polymer, and this is an unlinked monomer. So this is what we are seeing is a dehydration reaction in the synthesis of a polymer. So the water gets removed, and this gets attached, forming a longer polymer. And this is a vice versa case where you are seeing the hydrolysis of a polymer. So this is a chain, and then a hydrolysis occurs, adds a water molecule breaking a bond, and then you get two shorter polymers. You get back this structure. So these are just mere quick recovery recaps. Okay, so these DNA and RNAs are basically your polymers. So your adenine binds with your thymine. So how do they bind? So what happens is that you have an OH over here, and the phosphate group has a side chain oxygen. So these water gets removed from over here, and then what you get is a bond. So it's a phosphodiester bond is being formed, and then your two monomers join together, giving your TA. Okay. So this is not what is the hydrogen bonding. This is just how these nucleotides attach one after the other, okay, forming the helical structure. Okay, so now as you can see here, so this is a DNA strand, this is your nucleotide sequence, and then you have a phosphate group, then a CH2 H, and then from the 3' group you again that water gets removed off, and then it joins via this oxygen of the phosphate group of the next nucleotide, and then in this manner an entire sugar phosphate backbone gets. So this is a sugar phosphate backbone and these are your bases. So these backbone, sugar phosphate backbone remains constant except for your RNA, where in this place of hydrogen you have an OH. Okay, so this is an intestine, and then it gets attached in this manner by your phosphodiester linkages. So now coming to the concept of base pairing. So how does these actually base pair? So one of is your cytosine forms hydrogen bonds with guanine, and your adenine forms hydrogen bonds with the thymine, and or vice versa case occurs. So what is important here to know is how many hydrogen bonds are being formed between these two nucleotides. One is your T and A, so it is only two hydrogen bonds which are being formed. So if you look back into the structure, which of these I don't think I have a structure over here. So if we get back to where we have seen the structures, so which of these groups are involved in the formation of the hydrogen bond? So first we need to look is AT. So A forms double bond with your T. So which of the groups are involved in the hydrogen bond? So in the case of A and T, what we see over here, that which first you need to find out which of the groups are actually capable of donating their hydrogens and forming this hydrogen bonding. So if we see over here, we have an N which contains a lone pair, and we have an NH2 group. So these are the two which will be involved in the case of your adenine, whereas for thymine it is going to be your O and your NH. So what will be, in which pattern will they form? So your oxygen, so this group will be involved in binding with your this one of your hydrogen, okay, forming one hydrogen bond, and the next one would be your this hydrogen. So I'm marking it as square with this hydrogen, which is going to form hydrogen bond with this N. So these are the two hydrogen bonds which are being formed. So this has two. So this has two hydrogen bonds. Okay. And your C, that is your cytosine, forms, okay, for the uracil also we have the same groups, only there is a change in the side chain. So it is going to be this oxygen with this one of this hydrogen, and the other one is going to be this hydrogen with your this nitrogen. So for C we have three hydrogen bonds between C and your G. So as I was mentioning, the three groups for your cytosines having this oxygen goes and forms hydrogen bond with this one of this hydrogen. The next one is this nitrogen which forms, so giving it a square, so this nitrogen forms hydrogen bond with this hydrogen, and the last one is this oxygen which will form hydrogen bond with one of this hydrogen. Okay, so these are the three hydrogen bonds which are being formed, and in this manner the base pairing happens, and then the double helical structure or the double stranded structure is being formed. So you see, so these are the in different manners of hydrogen bonds. So your CG has 3 and your AT has 2 hydrogen bond. Therefore, we can say that the your DNA, which has greater number of C and G, that is the GC content, are more stable and have higher melting temperature. So as the stability increases, the melting temperature increases. So DNA strands having higher GC content are more stable than the having higher AT content. So, one might ask that how these are hydrogen bonds. These hydrogen bonds are basically not much strong bonds. So these are basically your weaker bonds. So how does only hydrogen bonds affect the stability? So it is not that only there are only few hydrogen bonds. There are a lot of hydrogen bonds, and so these hydrogen bonds are like one person working is one person working 10 times is better than is obviously less better than 10 people working together. So these a person is not strong enough, but if you give 10 people that small thing, so they will actually add to the become more and more strong. So in this manner, these small bonds, the hydrogen bonds, are actually increasing the stability of the entire biological molecules. So, and the sequence of the nitrogen bond is responsible for determining the code of the DNA. So this is all about your base pairing. So these DNA have a double helix structure. So these shows the two strands of DNA in the form in which the DNA is mostly present in the cell. So in our cells, we mostly get to find this bDNA. So bDNA structure is more naturally occurring. So you can see that the difference between these two is 1 nanometer, and the difference between your the pitch. So the pitch of a DNA is your 3.4 nanometer. Okay, so we call this the pitch of a DNA. Okay, so this is the pitch, and there is the difference between the two nucleotides is 0.34 nanometer. So if the pitch is 3.4 nanometer and the difference between the two is 0.34 nanometer, so how many nucleotides are present, or how many strands of how many these structures, these nucleotide structures are present? Tell me. So if the pitch is 3.4 nanometer and the difference between the two nucleotides is 0.34 nanometer, so how many nucleotides are present in this 3.4 nanometer? So there are so anyways, there are basically, so this is an easy question. So there are, okay, you people tell me first, then I am telling. Yes, so there are 10, but is it 10 because the difference between them is 0.34? Okay, and you see that it is so we what we do mostly is we divide it, but if you think it logically, that there are probably 11. Okay, because the difference between the two is 0.34, so the difference between the 10th and the 11th would give you the 10 one. But in anyways, this 10 factor gets correct. Okay, so there are 10, so it's 3.4 by 0.34 nanometer, so that is your 10. Okay, so now coming to the nucleic acid strands. So these nucleic acids are your polymers which are derived from your nucleotides. So the backbone consists of the repeated phosphate deoxyribose units. So you need to remember these things. And these individual nucleotides are joined by your phosphodiester linkages. So like proteins, your nucleic acids also have a polarity which is determined by the 5 prime and the 3 prime ends of the sequence. So basically how we write these DNA strands, or in this manner, it is conventionally written in the form of left to right, from 5 prime to the 3 prime end. So your 5 prime end, why we call this? Because your 5 prime CH2 has the phosphate carrying group, so it is the 5 prime end, and then they are linked by your different phosphodiester bonds, and your 3' group is determined by that 3' OH. Okay, so and it is written in 5' to 3' manner from left to right. And nucleic acids do have a polarity, which is because of the 5' phosphate group and the 3' OH group. So it is that nucleic acids also have a polarity. Now coming to the structure of the double helical DNA. So the cellular DNA exists mostly in the right-handed double helical form, that is your B DNA. So this is your B type of DNA, which is mostly found. And the double helix consists of two interwound antiparallel DNA strands. So one comes from the 5' to the, so the red one is the 5' to the 3' end, and the another one is your 5' to the 3' end in the complement. There is a 3' to the 5' end. Okay, and then the strands are complementary, but they pair together via your Watson-Crick base pairing model, that is your A to T and your G to C via the hydrogen bond. So AT has 2 hydrogen bonds and GC has 3 hydrogen bonds. So the backbone of these strands are located on the outer part of the helix, while the bases are stacked inside. And then they form major groups and different minor groups. So major groups are ones in which you see different, so these groups are like depressions or cavities, you can call. So major groups have bigger cavities than your minor groups, and these groups can be what you can be utilized for different drug treatments, and actually you want to see that whether the drug is binding to this group or not. So these groups can be actually applied in different applications and finding out in different drug trials and all. Okay, so in the most common conformer of the double helix, that is it's a BDNA. So about 10 base pairs occurs per turn, as I was saying, 3.4 by 0.34 nanometer. So these BDNA molecules contains wider major groups and narrower minor groups, in which the parts of the bases are exposed to the outside. Now DNA binding proteins locate and interact with specific base sequences which are exposed in the group. So as I was saying that different trials can be done, different proteins can be used, the DNA binding proteins can be used, which actually binds with these groups, which interacts with the specific base sequences exposed in these groups. Now what is DNA bending? So we know that DNA can be bent because it lacks strong, or you can call the stabilizing bonds, which are oriented parallel to the axis of the double helix. Now bending commonly occurs on binding of different your transcription factors like your Tata box, you have heard of it, that is your Tata box binding protein, that is the TBP, and the DNA binding proteins such as your histone. So bending, why is this bending important? So bending is crucial for the packaging of the DNA inside your chromatin, which are present inside your eukaryotic cells. So this DNA actually bends in the presence of your transcription factors. So there are different transcription factors which comes and are actually important in uh, what you say is packing of your DNA. So these transcription factors are basically your proteins, the different proteins. So like we have the TBP and your DNA binding proteins, like your histones and all. So these are crucial for the packaging of the DNA inside your the eukaryotic cells. So now coming to the supramolecular structure. So in many cases you have seen that your multimeric proteins have very large sizes, possibly your 10 seconds to 100 seconds of your subunits, and these complexes have a very high level of structural organization, which gives it a name of supramolecular structure. So some of these examples includes your mRNA transcription pre-initiation complexes, then you have the ribosomes, the proteosomes, and the spliceosomes. So these supramolecular complexes function as the macromolecular machines in performing the different activities of the individual subunits, which cooperates in the performance of different protein synthesis by the ribosome. So you have the RNA polymerase and the different transcription factors followed by the mediator complex. So it sits on the promoter sequence, and then the transcription pre-initiation complex is formed, and after this complex is formed, then the transcription is initiated and followed by the different protein synthesis by your translation. So these supramolecular structures are important in the different uh, functions and all. Now coming to the base catalyzed hydrolysis of your RNA. So your DNA and RNA differs on different aspects and all. Okay. And with so this difference, when this difference is with respect to the sugar which is found in the nucleotide monomer units, that is your 2 deoxyribose versus your ribose. So the phosphodiester bonds of RNA are susceptible to your hydrolysis in the basic conditions. And why is it so? Because of the presence of the hydroxyl group inside the ribose. So because of the presence of these 2 prime hydroxyl group, it becomes more susceptible, the RNA is more susceptible to your hydrolysis. What happens in contrast for the case of DNA is that the phosphodiester bonds are much less susceptible because it does not contain your 2 prime OH group, and because of this, it was thought that DNA was selected over RNA as the preferred molecule for the long-term storage of the genetic information, because it is less reactive molecule due to it's because RNA is a more is a less reactive molecule due to its containing of the 2 deoxyribose. So your RNA is more reactive and your DNA is less reactive, making them so RNA is more susceptible to your attack and hydrolysis, whereas your DNA is not that much susceptible. Okay, so this is the base catalysis of hydrolysis. So what happens is the OH comes and attacks this hydrogen, and then the bond breaks over here. So you get a 2 prime 3 prime cyclic monophosphate derivative, and after the attack of water, once again you get 3 prime monophosphate and the 2 prime monophosphate, because now you do not know whether the water is going to in attack over this or this. So depending on which side the water molecule is attacking, you get the different monophosphate, that is the 3 prime on the 2 prime. So this is about the base catalyzed hydrolysis of your RNA. So this also comments on why your RNA is less stable than your DNA, because its susceptibility to hydrolysis. Now coming to DNA denaturation and the renaturation. So we have heard a lot about this denaturation and the renaturation for the, which is called the folding and refolding of proteins. It is more over like that. So So your DNA denaturation refers to the unwinding and the separation of the 2 strands of your double helical DNA molecule. So inside the cell, that is in vivo, your enzymes such as your helicases separate the DNA strands. Now in vitro, the denaturation is achieved, can be achieved by heating. So when you heat the DNA, they opens up, and also by the treatment of low ionic strength buffers or extremes of pH. So in these manners, you can actually denaturate different. So what is important over here? In which techniques can you denature your DNA? So there can be questions like this: what techniques can be used inside the cell for, or which enzyme is involved in DNA denaturation inside the cell, or what factors are actually involved in the denaturation of your DNA. So there can be questions like this. So we need to know one of them is helicase, which is important, and then heating also denatures your DNA, and also your low ionic strength buffers or high extremes of pH can hamper your DNA. And this DNA denaturation can be monitored by your UV absorption spectroscopy, because your GC base pairs, that is your guanine cytosine, is held together by 3 hydrogen bonds, whereas your adenine and thymine is connected by your 2 hydrogen bonds. So the melting point of a double helical DNA increases with the GC content. So what happens in DNA renaturation? The double helical molecules are formed by the annealing of your single-stranded molecules. So this is when we come to the concept of your PCR. And in PCR we do annealing. First we denature and then we go for annealing. Okay. So what is important over here is that what factors actually denatures your DNA. That is number one. The number two is that how can you monitor your DNA denaturation? That can be monitored by your UV absorption, and then the higher melting point can be because of the higher GC content. So that is number three. And then your DNA denaturation, the double helical molecules are formed by the annealing of the single stranded molecules. So you go so in this graph what you can see is that this is an absorption at 260 nanometer, and then in the x-axis you have temperature. Okay, so if you see that this is a double stranded DNA, and then the temperature is being increased, and say your melting point temperature is at 85, so then it increases, so absorption increases with the single stranded DNA, and then it's constant, and at one point it just breaks open and forms the single stranded DNA, and single stranded DNAs have higher absorption. And when you plot the percentage of your GC pair versus your TM, then you can actually tell that with the increasing TM, this percentage means with the increasing percentage of your GC pair, the TM also is increasing. Okay, so these are the two graphs which also helps you to monitor the DNA denaturation and all. Now coming to DNA supercoiling, so the processes such as your replication that actually unwinds your double helical DNA introduces a kind of a torsional stress that results in your supercoiling. So this is most evident in your circular DNA molecules like your bacterial plasmids, sometimes your viruses, but occurs in linear eukaryotic chromosomes as well. Now what happens is that in replication the overwinding happens ahead of your strand separation site, and there is some kind of positive supercoiling. Now underwinding also happens, which occurs resulting in your negative supercoiling. So these are the different directions in which your coiling is occurring. So these are the different supercoilings. So enzymes such as your topoisomerases regulates how much the supercoiling will in DNA is going to occur. So there are two types of topoisomerase, actually three. So two are basically very important. One is the topoisomerase 1, which relaxes your supercoiled DNA, and your topoisomerase 2, which introduces your negative supercoils inside your DNA. So the cellular DNA naturally exists in a slightly negative supercoiled state, and this negative supercoiling facilitates other processes such as your replication, then transcription, where your DNA is unbound. So what we need to keep in mind about this DNA supercoiling thing is first that processes like your replication introduces the torsional stress, because of which your supercoiling is occurring. That is number one. So where do you observe this supercoiling? It can occur in your bacterial plasmid, sometimes your viruses, and in sometimes in your linear eukaryotic chromosomes as well. So there are two different types of supercoiling. One is a positive and the negative supercoiling. So the next thing is that what enzyme is involved in regulating the amount of supercoiling in DNA, that is your topoisomerase. And there are two types of topoisomerases. One is topoisomerase 1 and the other one is topoisomerase 2. So topoisomerase 1, what it does, it relaxes your supercoiled DNA, whereas the topoisomerase 2, it introduces the negative supercoils inside your DNA. And so that's all we all need to and so if you can see from this figure is that this is a supercoiled DNA and this is a relaxed one. You can see that there are different loops and this is being formed. Okay, right. Now coming to the RNA structure. So we see that most cellular RNAs consist of a single strand. However, there are double helical regions which are common in RNA where the complementary sequence region occurs. That is, it folds back forming hairpins or stem loops. You see complementary sequences and they come over each other forming these different types of secondary structures. So the common types of RNA secondary structures are your hairpins and your stem loop structures. So the stem regions forms a DNA type double helices. Now when these turns are connecting the helices, these are shorter in hairpins than your stem loops. So RNA also forms 3D structures in molecules like we see in your ribosomal RNA or your transfer RNA. So the pseudoknot which is being found in the human telomerase RNA3. So these 3D structures are of these ribosomes are rivals those of proteins with respect to complexity. So what we need to know is that how these secondary structures are being formed and what are the different types of secondary structures which is being formed. So one is your hairpin structure and the stem loop structure, and the turns connecting the helices are basically shorter in hairpins than that of your stem loops. And the third is that how the structures, these tertiary structures are being formed due to this pseudoknot and all. So we just need to keep these things in mind. And the other important thing is that these stem regions forms a DNA type double helical structures. So that's all. So what do we now require this DNA for? So one is that it helps to detect, enumerate clone genes. One is of detection and enumeration of your species. Then detection and sequence specific, identifying and detecting the sequence specific DNA regions, and also creating different DNA constructs, which is known as your RDT or recombinant DNA technology. And how about RNA? So it helps us to find the answers to which genes are being transcribed and what is the location of these genes as to when and where these genes are being transcribed and what is the level of transcription that is occurring. So these are the questions which helps us to answer these are the questions which we are looking the answers from when we ask a question like what do we need DNA and an RNA for. So just because the DNA stores your genetic information and the different uh, information about your genes, so you can actually do different recombinant DNA technological things, and then identifying the different regions where your DNA is going to bind and all, and then it helps in the cloning and detection and enumeration of your species. But when you study about RNA, it is about what is the level of transcription that is occurring and which are of the genes are being transcribed and where is the location of such of these genes. So different comments can be made when we study DNA and RNA. So coming to the next thing, that is your DNA purification. So what we do, a culture of the bacteria is primarily grown and then harvested, and after that we undergo some kind of centrifugation and then we pellet down the cells. So the cells are removed and broken to give us a cell extract. That is, the process is known as cell harvesting and lysis. So we get that cell extract, and then we go for DNA purification. So the DNA is purified from that cell extract which we have got, and after that the DNA is concentrated, and then we check its value at a 260 by 280, and then we check the ratio as to how pure our DNA is. So first we go for cell growth, then cell harvesting and lysis, then DNA purification, and then the DNA concentration. So the cell extract is what we are looking for. Now, bacterial genomic DNA preparation from the cell extract. So first is your cell lysis, where you disrupt the cell wall and the cell membrane, and you collect the cell extract. So this lysis of this disrupting of your cell wall and your cell membrane is known as the lysis, where you use different things like your you can use detergents, then your organic solvents. Sometimes we use, so mostly you use lysozymes and heat. So these different ingredients can be used for the lysis of your cell. So lysis of your cell is disrupting of the cell wall and the cell membrane to collect the cell extract, and then by centrifugation you remove the cell debris from over there, and then you collect the cell extract, from which you can get your DNA, RNA and protein. So then you can find out what are you actually looking for. Is it the DNA or the RNA or the protein? So your cell will contain all of these. Now after this, so this cell extract contains your DNA, RNA and your protein. So what you do is you mix it with phenol, and then separate the layers by centrifugation. So your aqueous layer is going to contain your DNA and the RNA, whereas your interface will mostly contain your coagulated proteins. Okay, and then in the bottom you have your you're going to have your phenol. So this is your phenol chloroform method of separation. So you need to basically gently mix it, because to avoid the shearing breakage of the genomic DNA. So adding the enzyme RNase can actually help you degrade the RNA in the aqueous layer. So first you get the cell extract, where you would be having your proteins and your RNA. Yes, your bottom can have cell debris. You need to filter that out and use it. And then you separate, and in this way, from the aqueous layer, you add RNAs to degrade the RNA in the aqueous layer. And from after that, you filter it out, and then you get the pure form of your DNA. So there are two ways in which you can concentrate your genomic DNA. One is by spooling. So what we do, we have the concentrated DNA solution, and above that we have ethanol. So these are the DNA fibers which we use, and the glass rod. And after the method of spooling, we concentrate the genomic DNA thing. The other form is that the DNA which is being precipitated is collected by your centrifugation. This is known as the ethanol precipitation technique. Okay, by using ethanol, you precipitate down your DNA, and then you collect them by centrifugation. So the concentration of ethanol being used over here is 70%. So ethanol plays a very important role in precipitating your DNA thing and all. So we know that in plants, so how can we get rid of these carbohydrates? So genomic DNA preparation in plants. So you have your cell extract, and then we have a cationic detergent, CTAB, and after that we go for centrifugation. And this when we are adding a detergent, your nucleic acid is going to precipitate out, and after forming that nucleic acid CTAP complex, and in the aqueous solution you're going to basically have your carbohydrates, the sugars, the carbohydrates, and your proteins. And after that, what you do is, when you know that the supernatant is not of any use now, so you discard it because your pellet has the nucleic acid C type complex. And then what you do is resuspend the pellet in 1 molar of your NaCl. Okay. And then what you do is you perform the same thing, that is your ethanol precipitation and ribonuclease treatment, and after that you get your pure DNA. So here two things are important. One is your CTAB, and then the other thing is that you go for ethanol precipitation. So next thing is about plasmids, which we know as the vehicles of recombinant DNA. So these are our plasmids. Here we have the genomic DNA. This is a bacterial cell where we have the genomic DNA and the plasmids. So what are these plasmids? These are non-chromosomal DNA, and here it goes replication, which is independent of the chromosome. So there are many copies per cell, and these are basically very not very. These are basically easy to isolate and manipulate. So how do we purify these plasmids? So these are the supercoiled plasmids which we supercoiled plasmids which we have, and then these are some of the linear DNA. So when we give it a pH of 12 to 12.5, we get single-stranded linear DNA because it gets unwinded and it opens up at extremes of pH. We have just heard, so alkaline conditions to denature your DNA, and then we give it a pH of 7 where there is tangled mass of linear DNAs, and after that what we do, we centrifuge it. So and then the pellet of the linear will contain your linear DNA molecules, whereas the supercoiled plasmids will remain in your aqueous solution. So from this you can actually separate out the two and then undergo your plasmid purification. So other thing is that the genomic DNA can't renature, plasmids can, because they never fully separate. Now coming to the next type of DNA purification. Okay, we will talk about this in a few minutes. Okay, so let's take a 2 minutes of break, after which we can start. Okay, so let us start once again. So the next way of how we can purify our DNA is by silica binding. So what happens is that we have a matrix of silica, and then the DNA molecules goes and binds to these silica particles. So this binding occurs in the presence of high salt concentration, and you can disrupt this via your elution. So you will see that there are different columns which has your silica particles, so it binds by this. Also you have different buffers for this elution. So what this elution buffer does, these are very low concentration. The elution buffer has very low salt concentration, mostly your water, but some for long-term storage we actually use elution buffer for its DNA purification, but mostly water can also be used for the short-term storage and all. So during DNA purification occurs, so DNA has a tendency to bind to the silica particles. So these DNA purification can be done by your column chromatography, where you have the silica particles and then you pass your DNA and then you elute by water. So when you are washing it, the denatured, so you have a wash buffer as well. So in that wash buffer, what you do, you have the denatured biochemicals and all the other discards which actually comes down when you are washing it. Then that is what you are doing, is you are cleaning up your DNA, but it does not elute. When you actually put water and keep it for some time and then go centrifuge it, what you get is the DNA. So in this manner also you can purify your DNA via the silica binding technique, and mostly in labs and all you will be seeing this technique where you would be purifying your plasmids, then your PCR products, the gel extraction products, where you have the DNA and you need to purify your DNA. So this is the most commonly used technique you would be mostly observing in the laboratories. So the other way, as I was speaking, the phenol chloroform extraction, you have 1:1 phenol chloroform, or your 25:24:1 phenol chloroform and isoamide alcohol. So what these chemicals do, or what these organic things do, is the phenol is involved in denaturing your proteins and precipitates form at the interface between your aqueous or the organic layer. The chloroform increases the density of the organic layer, whereas your isoamine alcohol prevents the foaming. And by using all of these, you can actually purify your DNA. So if you're asked, you can be asked questions about the functions of these different organic solvents. Okay, like what is the role of phenol, or what is the role of chloroform? Okay, so you can be asked these questions. Then you have to say that phenol is involved in the denaturing of your proteins, and the precipitate forms at the interface between the aqueous and the organic layer, whereas the chloroform increases the density of the organic layer. Okay, so you have to answer it. So this is the same as that I have spoken before. So where you harvest a cell and then centrifuge it, you get a pellet of the cells. Uh, there can be other the unnecessary ingredients, and then you collect the cell extract, and after which you purify the DNA, and then you concentrate your DNA and you get the pure form of the DNA. So what is the problem which we face with RNA? So one is that your RNA is chemically unstable. The spontaneous cleavage of the phosphodiester backbones via your intermolecular transesterification occurs, which makes them, so these 2' OH makes them susceptible to your hydrolysis, because of which your RNA is chemically unstable. Also, your RNA is susceptible to nearly ubiquitous RNA degrading enzymes like your RNases. So these RNases are released upon your cell lysis. They are present on the skin and are very difficult to actually inactivate, because the disulfide bridges which are present over there, it adds to the stability, and also it does not require any kind of divalent cations like your magnesium or calcium basically for its activity. So because of this, this RNA is very stable, and we find very difficult to inactivate this, and this RNA Nases are involved are your the RNA degrading enzymes. Okay. Um, so this is are a few problems which we basically face with RNA. Now coming to another thing and a technique, which is your PCR, that is a polymerase chain reaction. So it was first described in the year of 1971 by your Kleb and Horana. We described and successfully performed it in the year of 1985. So this allows massive amplification of specific sequences that have defined endpoints, and this is a very fast, adaptable, simple and a powerful technique which allows us to apply it in different manner, in different ways, and it has a broad field of applications. Okay, because of this the PCR is very famous in the laboratories. Okay, and in forming the different clones of your plasmids and all. Okay, so why amplify specific sequences? Why is it important? So one of it is to obtain the material for cloning and sequencing for your say your in vitro studies. It also helps us to monitor if the gene expression, diagnose different kinds of your genetic diseases. It also helps us to verify and identify different engineered DNA constructs. Like I was speaking for the DNA RDT, that is your recombinant DNA technology. It helps us to identify what type of DNA constructs have we engineered. Okay, so amplification plays a very important role, and it also helps to reveal the presence of a microorganism or an entire individual. And there are different ways. So so these are the reasons why amplification is very important, and specific amplification of specific sequences plays a very important role. So what do we need for these polymerase chain? So PCR is polymerase chain reaction. So what do we need for it? It's a template DNA that contains our target sequence, that is the specific sequences that which we are looking forward to. It includes your primers, which so what are primers? These are short oligonucleotides that defines the ends of the target sequence, as to which particular sequence are we looking to amplify. So based on that we design our primers and which part of the sequence which we want to amplify. So that is important, and in that manner we design our primer and all. And all that we require is buffers, DNTPs, a thermal cycler. So a thermal cycler is one where we can actually change the so it's a cycle which we perform. So it's a polymerase chain reaction. Okay, so we require a cycler which is going to change the temperature and modulate the temperature. So first is the denaturation step, and then your annealing occurs, then the joining occurs. Okay, so so these are different temperatures they occur. So this is actually controlled by a thermal cycle which actually controls the temperature. And lastly we require is a thermostable DNA polymerase or the TAC polymerase. Sometimes we use the other enzymes like your fusion, which is going to play an important role in actually this PCR reaction. So this can handle very high temperatures, and because of that we use these particular enzymes in your PCR reaction. So these are the different things which we require. One is your template DNA containing a specific sequence. The next are the two primers, that is which will define the starting and the end point, as to which part you want to anneal. Then your thermostable DNA polymerase, different buffers, and your DNTPs, and finally a thermal cycler. So how it works? So simple PCR reactions. In the first cycle, you have a double-stranded DNA target. It can be single-stranded as well. So then the denaturation step does not occur. It just stays like that. So in for a double-stranded case, the denaturation occurs, and then followed by your primer annealing. So then the annealing is being done, and after that the extension step occurs where your DNA synthesis happens, and then denaturation by heat followed by the primer annealing and the DNA synthesis, and in this way the cycle continues. We have three cycles. One is your denaturation, then we have the primer annealing and the primer extension via your DNA polymerase, that is the thermostable DNA polymerase or the TAC polymerase. Okay, so you need to remember that there are three steps: denaturation, annealing, and extension. And after that, it is same as that. So the same thing continues. So now in the first case you had one, and then you get two, and this two undergoes the second cycle, then it undergoes a third cycle, and you get so many numbers. So usually we allow it to go for 30 cycles. So first the denaturation, then your annealing and your extension. So this step is actually done 30 number of times. So in the first case you have one, then you have two, then you get four, then 8, then 16, 32, and after your 29 cycles you get somewhat like a big figure like 134217, like that, and after 30 cycles you get even a larger number of that. So it's that into that. Okay, so this is the entire of the PCR reaction. So okay, so what you need to remember from here is that the denaturation occurs at a very higher temperature. So what do you do? So first you have your so I'm telling how we do a PCR. So first we have, say you are setting up a 50 microliter of your reaction mixture. You wanted the reaction mixture to be of 50 microliters. So first you know the DNA concentration of the, you know the template DNA concentration. Say it is around your 25 25 mg per microliter or micrograms per microliter. Okay, and then you add, or maybe it is around your, say uh, yes, around 20 micromolar or something like that. Okay, and then you add 2 microliters of it to make it around 50 nanograms and all. Okay, and then you add some amount of your DNTP, then about you get, what you get is a 10x sorry, a 5x buffer. So you add 10 microliter of it in your 50 microliter solution, and then you add the DNTPs, then your primers, depending on what is the concentration of the primers you have. Then in the after you have mixed all of these ingredients, you mix them very gently, and then in the end you add some amount of your enzyme, which will actually perform, and then the entire mixture is put in the thermal cyclone. And then at firstly in the first it will start denaturing at higher temperature, and then the annealing occurs. So this annealing temperature is depends on the melting temperature of the two primers. So if the melting temperature of your primer is say 70 degrees centigrade, so what would be the annealing temperature be? That is one question I'm asking to you people. And what if so if your two primers have an melting temperature of 70, that is one question. And the other question is, say your melting temperature of the two primers are 70 and 68, so what would be then the annealing temperature that you are going to use? And then after that you perform the extension, which occurs depending on which in which of this temperature these enzymes actually function properly. So depending, like your fusion enzyme works at 72 degrees centigrade, whereas your TAC occurs in between 70 to 72 only. So 72 is the optimum one. Now tell me if the say I'm asking you only the first question, that if your primers have a melting temperature of 70 degrees centigrade, then what is the uh, what will be the annealing temperature that you are going to use? Or how do we calculate the annealing temperature? Because because when we order the primers, we actually check the Tm of, or the melting temperature of these primers, and depending on that we set the annealing temperature. So What would be the annealing temperature if the Tm is or if the Tm of your primers are 70? Tell me. No one? Okay, see. Yes, the annealing temperature has to be less than the melting temperature. That is true. So, how much less it is that? So, usually we take 5 degrees less than that of the melting temperature. Now, see, there are obviously we need to optimize these things. Not always it is that the temperature has to be 5 less than your annealing temperature. It can be even more or less, so we need to optimize it, but the optimum one is that your melting temperature has means the annealing temperature has to be a 5 degrees less than that of the melting temperature, and the other things remains as it is. Now coming to the questions. So that is all with the lecture and all. So now questions. So these are very easy questions I have kept, and I am expecting that there would be easy questions also from this part. So the first question says, to lyse the bacterial cell wall for extraction of DNA, which of the following reagents is commonly used for lysing the bacterial cell wall? A, yes, that is the right answer. We use lysozymes because lysozyme is involved in disrupting the bacterial cell wall or the cell membrane. Right. The next question is, isolation of DNA plant sources poses challenges due to the presence of. Okay, so as I was teaching about the carbohydrates and the different metabolites and all, sometimes proteins. So yes, so the presence of polysaccharides and secondary metabolites makes it difficult to isolate DNA from the plant sources. But there are different purification techniques which we can actually use for purification of the DNA from plant sources. But in anyways, 2C is right because of the presence of the carbohydrates, polysaccharides, and the different secondary metabolites. Now coming to the third question, the function of EDTA during cell lysis is. So what does EDTA do during cell lysis? Is it that it acts as a metal chelating agent or inhibiting cellular enzymes that degrades the DNA, or it destabilizes the cell wall, or all of this? What is the role of EDTA? A, it acts as a metal chelating agent. Yes, that is right. It acts as a metal chelating agent, but it also is important in destabilizing the cell wall. It destabilizes the cell wall, and it also inhibits the cellular enzyme that degrades your DNA. So all of them are actually correct. Yes, all of the above is correct. Okay, so the next question is, what is the function of NaOH in lysis buffer? What is the role of NaOH? This is an MSQ question, one or more than one can be correct. Okay, so if you think more than one option is correct, you can tell actually. Remove proteins from the cell lysate. Okay. Do you think anything else? Actually, the role of NaOH is to denature your genomic and plasmid DNA. Okay, and it also breaks down the cell wall. So So your function of NaOH is to denature the genomic and the plasmid DNA and breaking down the cell wall. So your both A and B is correct. D is not correct because it does not help in removing the proteins from the cell lysate, and also it does not denature your RNA. Okay. Now coming to the next question, one is that RNase A degrades your RNA that are present in the cell lysate. What will happen if RNase A treatment is not done during our DNA isolation? So what is going to be the problem? So what problem are we going to face? So both RNA and DNA will form complex and precipitate out of the solution. You said this. Okay, that can be one possibility, but you see that we are not providing any medium or a chemical or an organic solvent because of which it's going to precipitate out. Okay, so the precipitation is, it is true that, but after we are giving it some kind of treatment, that is possible. But here we are asking that RNA degrades RNA. So what will happen if RNA treatment is not done? So the RNA will be present. So RNA will not be removed. And as your DNA and RNA both will absorb at 260 nanometers, so you would not be able to actually understand whether you're getting the absorption for the DNA or the RNA. So that will actually uh, cause a different kind of problem during the quantification of your pure DNA. So question number 5B is the right option. That your RNA will not be removed and both will absorb at 260 nanometer, and it will find you will face difficulty in quantification of the isolated pure DNA. Okay. Next question: what is the role of SDS in lysis buffer for plasmid DNA isolation? Yes, so SDS solubilizes the membrane, it denatures the protein, and it also disturbs the DNA protein complexes. So you say all of this, yes. So 6D is the right. Okay, go for question number 7. What is the purpose of? Yes. So the purpose of plasmid isolation is, yes, one is we do it for cloning. One we do for gene expression, and it also acts as the vehicle for delivering target DNA in another organism. Okay, so yes, all of the three options are correct. So it is right. So which is 7D is correct. Isolating plasmid DNA from genomic DNA is based upon question number 8. See size difference. Do you know about the size of the plasmid or the genomic DNA? So what we can do is, what we know over here is that your plasmid DNA and your genomic DNA. So this question is mainly on the differences of the two types of DNA. So one in which these two differed is your conformation, and the other one is your charge difference. So these two would be actually important when we are isolating your plasmid DNA from the genomic DNA. Okay, so for question number, okay, sorry, yes, your conformation will vary and your size difference will vary, not the charge. Yes, so A and C both are correct, right? There is no charge difference actually playing a role over here. So size and conformation is correct. Okay. And okay, number 9, the neutralizing buffer used in plasmid isolation contains. What does the neutralizing buffer contain? Yes, correct. Why? Because the other three will actually break apart your DNA. So that will actually hamper your DNA, and it cannot be used for isolation. Okay. When neutralizing buffer is added after lysis to the cell lysate, what happens? No answer. Okay, so when the neutralizing buffer is added after lysis to your cell lysate, then what happens is that your chromosomal DNA gets tangled and it precipitates out. Also, your plasmid DNA anneals and remains in the solution, and the proteins and SDS forms your insoluble precipitate. So all of these are actually correct. Okay. Now when DNA bound to silica membrane can be eluted out by the addition of. It is a very easy question. Yes, water, I just explained. To wash DNA bound to silica membrane, which of the following reagents are used? How do you wash silica membrane? B, it is DNA. Then it is, you have been asked to wash the DNA bound. It's about washing, not about eluting. When you use water, it will actually elute. Yes, it's deionized water, but for washing, what do you use? You do not use detergents because it will then hamper your DNA. You do not use any kinds of other salts because it will again make it tighter, and you do not even know whether a pH of 8 or not suits your DNA or not. So we use ethanol or isopropanol for. So you will see when you have when you actually wash, then or you use different kinds of kits for DNA purification and DNA isolation. You will see that you get a very little amount of a solution, and you have been asked to add some amount of your isopropanol or ethanol into it. After that, you can use that solution. So what you add over there is the 100% ethanol. Okay, so then there is some solution that is some amount, and then you actually add ethanol to it, and then you make it a wash buffer, and that wash buffer is actually used for washing your DNA bound to your silica membrane. So you basically use ethanol or isopropanol. Okay. Okay, the next question is quality of the purified plasmid DNA has to be checked by. So how can you check how pure your plasmid DNA is? By UV spectroscopy. Okay. Okay. So we do not actually use size exclusion chromatography, that is for proteins, and we do not run page usually. Okay, but we use agarose gel electrophoresis while studying of DNA. So I was teaching in my previous lectures as well about the agarose gel electrophoresis, where you have ETBR which intercalates inside your DNA strands, and because of that your DNA fluoresces. So you can use UV spectroscopy is absolutely correct, but you also use agarose gel electrophoresis. So after you run your DNA in that, you see whether you're seeing any other bands or not. And if you see only one single thick band, then you can tell my DNA is pure. So that also helps to comment on the purity of your DNA and all. Okay, so both B and D are correct. The next question, the quantity of the purified DNA can be estimated by measuring the absorbance at. Yes, 260 nanometer. We check at 260 nanometer for the nucleic acids and 280 nanometer for the proteins. Okay, so is correct. These are easy questions. In agarose gel electrophoresis, the plasmid DNA runs ahead of the linear DNA. Why? Why does your plasmid DNA run ahead of the linear DNA? Yes, because your plasmids are supercoiled, which helps them to migrate in a faster rate because they are more compact, and because of that it will migrate faster. Yes, that is correct. Okay, so that's all with from my part about the question and answers and the lecture. I think I'm done from my part. Thank you. If you people have no questions, I'm done. Any questions here? Okay, that's good. Thank you then.